this is where we started before, but I didn't want to give you the answer to the next question. So we know that this one is regeneration. Okay, our next question. Which provides the strongest evidence for the relationship between arthropods and roundworms? Now, we really haven't talked about this, but an arthropod is going to be arthropoda, is the phylum where we have our crustaceans and our insects. So our roundworms now are going to be our nematodes, which are our parasitic worms, heartworm, whipworm, those kind of things. So shared anatomical features does not fit because they don't look anatomically anything alike. Shared functional characteristics, they don't function the same either. We've got one that is totally parasitic and one that is not. Embryological development cannot be it either. So for this one, your answer is C, similar protein structure. When we made our proteins from our amino acids, we said that that was a universal code, that all of those amino acids, the proline, the lysine, the valine, all of those we put together were going to be the same whether it was bacteria or whether it was us. So the answer to this one should be C. Yep, there we go. All right, looking at this animal, which is the dorsal surface of the hummingbird? Now this is showing you the picture that we had in our PowerPoint. And when you have cephalization, which is forming a head, you have these demarcations such as anterior, posterior, dorsal, and ventral. If you ever get confused, think of the dorsal fin on a shark. The dorsal fin on the shark is the fin that you see above water. So we know that A is going to be anterior. We know that C is going to be posterior. So B is actually showing the backside of the hummingbird. D is showing the belly side. So this answer would be B. All right, which organism exhibits cephalization? Now they're asking you which one has the formation of a head. Well, we know that a jellyfish doesn't have a head. We know that a sponge doesn't have a head. Starfish don't have a head. So I think our obvious answer here is B, snail. All right, how is deuterostome development different from protostome development? All right, well, we know that protostomes, that opening into the gastrula becomes the mouth. Proto means first, stoma means opening. Deuterostomes, deutero means second. So the blastopore in the gastrula becomes the anal opening. All right, so look at our answers, and we've got our picture that shows that we have the blastula at the top. The next is going to be the gastrula, where we have endoderm, ectoderm, and then we have the mesoderm there. And then it shows you, these are cross sections, that you have the gut in the middle and the mesoderm, and then we have our coelom. Okay, so let's look at our question. How is deuterostome development different from protostome? A blastula does not form. Well, we know a blastula does form. The coelom is less advanced. The gut is lined with mesoderm, or the blastopore becomes the anus. Which one do you think it is? The answer to this one, even though in deuterostomes the blastopore becomes the anus, if you're looking at this picture, that's not what it what the picture is depicting. The picture is actually showing you that the gut is lined with mesoderm, so I believe the answer to this one is C. Okay, the gut's lined with mesoderm. That was a little bit more difficult. That one made you think. All right, why are sponges most distantly related to the rest of the animals? They're not multicellular. Well, that's not true. We know they are. They do not have tissues. That's true. Their embryos have a mesoderm, or they arose from a different protist ancestor. Why are they most distantly related to the rest of the animals? They don't have tissues. We know that they are multicellular. Their embryos, mesoderm doesn't apply because they don't have tissue development. So your answer is B. What is the term for organisms that remain attached to one place? This should be easy. The only other one, tactile means touch, and axillary does not apply either. Sedentary just means you're still. If you're sedentary, maybe, you've been sitting on the couch watching TV, you're sedentary, you're not moving around. But when you're permanently fixed to a surface, we know the answer to that is A. Okie-doke. What provides the energy for the discharge of a cnidarian's nematocyst? Muscle fibers, hydraulic forces, osmotic pressure, or protein springs? I don't even know what a protein spring is. So which one of those do you think? The only two that you could possibly narrow this down to be is either going to be B or C. The reason muscle fibers don't apply is because cnidarians have endoderm and ectoderm, but in order to have muscles, they would have to have mesoderm, and we know that cnidarians don't have that. Now, I'm probably going to ask it to you as hydrostatic pressure, but that's not the same as hydraulic. So a hydraulic force and hydrostatic pressure are completely different things. And if you chose B, know that it would never end with force. It would always end with pressure. Okay, which is the dominant stage in the life cycle of a sea anemone? Bud, hydroid, medusa, or polyp? Which one do you think it is? Which one? Okay, so bud and hydroid are really not applicable. Bud is a new organism from budding. A hydroid is going to be a type of jellyfish that lives in colonies like your Portuguese man of war. So now we're down to the medusa and polyp. Which one do you think it is? Okay, now the reason that this is the dominant stage is because polyps are fixed. Now don't confuse dominant with the adult stage, because the adult stage in a lot of jellyfish is the medusa. But our very first phyla, which is Anthozoa, if you remember, all of them are always in the polyp form, and that's our sea anemones and our corals.